And we might as well get started. So it's a great pleasure, and I'm really excited to welcome Luiz Pessoa today to our lecture in the SFB 936 of our consortium. Let me do a brief introduction. So to many of you, Luiz may be familiar from interactions on social networks, from his really very impressive research output, and from, in particular, some of the reviews who have really elucidated some of the very complex relationships in our field in cognitive neuroscience, but also in network neuroscience. Just a little bit of background. So Luiz Pessoa received a BSc and an MSc in computer engineering and computer science from the Federal University of Rio de Janeiro in Brazil in 1989 and 1990, respectively. He then followed up by doing a PhD in computational neuroscience at Boston University in 1995. Then he became part of the faculty of computer science at the Federal University of Rio de Janeiro again, before once again returning to the US as a visiting fellow at NIMNIH with Leslie Ungerleider in the Laboratory of Brain and Cognition. That was until 2003. And then he worked his rank through increasingly more prestigious faculty positions, first as an assistant professor at Brown University in the Department of Psychology, at the Department of Psychological and Brain Sciences at Indiana University in Bloomington, as an associate professor, and he's now at the Department of Psychology, University of Maryland, College Park, which, as he states, is close to DC, where he is a professor and director of the Maryland Neuroimaging Center. His interests center around the interactions between cognition and emotion and motivation in the human brain. And as well, there's an additional focus of his research on the development of statistical and computational tools for the analysis of fMRI data. He's produced at least 2 books that I'm aware of: The Cognitive Emotional Brain back in 2013, and most recently, The Entangled Brain in 2022. And we're internally in the lab, we're joking that there must be multiple clones of him because, I mean, both by the research output and by his presence on social networks, on Twitter, and now also on Mastodon. It's 24/7, and it's always stimulating to see your comments, to see your inspirational tidbits. But I also want to highlight that he's a great popularizer, in the very best sense of the word, of science, of the very complex relationships that exist in network neuroscience and cognitive neuroscience. And it's in that spirit that he also presents his lecture today on the entangled brain, the integration of emotion, motivation, and cognition. And I'm very much looking forward to that.
Thank you, Klaus. That's a very, very warm, wonderful introduction. I should ask whether you would rather have the questions during the presentation or at the end. Any format works for me. I just need to probably keep up with the raising hand, or or just wave there somehow. But whatever way you want to handle questions is fine with me. Do you have a preferred way, or?
So typically we have the more substantial discussion at the end, but I guess if there are technical interludes, then, and you're happy to break for them.
I'm happy. Yeah. So if I don't seem to be reacting, please wave or something, because I might be missing the feedback here. Okay. All right. So let me get started. So historically, we've been interested in understanding the relationship of emotion in the brain and how it's actually instantiated. Common idea that has been very influential in the literature for the past 80, 100 years is that emotion involves separate and independent neural circuits. This came to a really clear-cut proposal with MacLean in the late 40s and early 50s, with the proposal of the limbic system, or the visceral brain, a set of structures centered on the hypothalamus and the hippocampus that were supposed to be sort of the center of this emotional brain. If we fast forward to today, the circuit definitely looks different, but the notion that there's a dedicated system for emotion is what still plays a central role. So the details have changed. Now we might be interested in regions such as the orbital frontal cortex shown here in blue. Perhaps it includes the anterior temporal pole, the anterior insula. It has regions such as the amygdala which weren't present in the very early versions. But nonetheless, we have an emotional brain, a set of dedicated structures that provide the substrate for emotion in the brain. Even more importantly, in this idea of having a dedicated circuit is the idea that within this circuit, there has always been the notion that there is a region, a specific region, area, structure that plays a disproportionately important role. So we can see that by going back to the 1920s, even 20s and 30s, and all the way up to the 50s, in where the hypothalamus was really at the core center of this conceptualization of the emotional brain. And it is perhaps in the past 30 years or so that this has shifted, and now the center seems to be, the epicenter of the system seems to be the amygdala. So if we take that into consideration, there is this notion that there's a clear-cut emotional brain. There's a system that is dedicated for emotion. But one of the things that I've been very interested in, and Klaus for quite some time as well, done wonderful work in this line of thinking, is that we can consider anatomical connectivity from a computational perspective and look at general patterns of this anatomical organization and try to understand what kinds of functions it might support in more natural or more difficult ways. So one of the things that strikes me is that when we look at anatomical connectivity, for instance, even as simple as anatomical connectivity, even in early incarnations of these diagrams, one of the first of them from Young and colleagues from 1994, and we have many, many others much updated in the last decades, that we might be looking at this problem from the wrong angle, in the sense that having a circumscribed center for emotion turns out, given this neural architecture, to be quite a difficult proposition, because the amygdala, for one, is a structure that is massively interconnected. In particular, the basolateral amygdala is massively interconnected with large portions of cortex. And in fact, there's a paper, even from 2001, that Larry Swanson and colleagues estimate that the amygdala might have 600 to 1,000 connections from the assessment that they were trying to do at the time, even as long ago as 2001, that it might have as many as 600 to 1,000 connections spanning cortex and subcortex. And this property of this kind of massive amount of connectivity is not something that is restricted to the amygdala. The amygdala doesn't happen to be enormously unique in this respect. In fact, there's a version of computational analysis of large-scale connectivity in monkeys that was performed by Moda and Singh and published in 2010, in which they look at the entire neural axis. I think they were not going all the way to the cerebellum, and I can't remember how much of the brainstem they were covering. So, but a considerable amount of the brain was included. And what came out of their analysis, and there are many ways of looking at this, but there's converging evidence from other kinds of analysis. What comes out of the analysis is that major sectors of the brain, including the major lobes, temporal, insula, cingulate, parietal, frontal, and so on, thalamus for one, the basal brain, including the amygdala and other structures in the striatum, are really massively interconnected with each other, such that there are pathways linking these different sectors of the brain that only take a few anatomical steps. And this anatomical connectivity is so dense that in their figure for their paper in PNAS, I think they applied something like a 30% density of the connectivity, because if they went up to 70%, for instance, they just became too much and you couldn't even see anything. So if we step back from this, it has suggested to some people, and to me in particular, the idea that we should be thinking of, instead of a framework in which it tries to isolate subsystems, to try to focus on frameworks that try to integrate. So we might have something like perception and perceptual competition. Attention plays roles into selecting what items survive this competition. You might have executive control and other aspects of competition that prioritize certain kinds of information versus other kinds of information, following this rule now versus some other rule. These are highly interacting anatomically and functionally. But what I suggested, and this is something that has been suggested before by many investigators that have studied perception and cognition, but one of the suggestions that we make is that it would be interesting as well to incorporate processing that is important for effective significance. And this was for this proposal, for the purposes of this talk, and just considering here sort of aversive negative stimuli, negative type of processing. And we can also consider the appetitive end of this situation, where you would like to approach and attain rewards, some set of processes, circuits, and so on that are important for motivational significance. So the integration framework that I've been wanting to develop puts this all together and trying to see, not that there's an arbitrary link between any two things, any two processes, or any two structures, but based on this highly organized anatomical backbone, what are the kinds of interactions and integration between systems that we tend to think as more or less separate does it support? So to look at that, we have, within this framework, developed a series of experiments that have tried to probe that. So let me give you two examples today. One is in the area of affective vision. How does emotional content shape visual perception and brain responses? So this is something that we worked for quite a while. I'm just going to give you one example. The example that I'm going to give you is this phenomenon called the attentional blink. The attentional blink, you can think of the attentional blink as a clip of 2 seconds or so, each frame of which lasts for 100 milliseconds, and they come one after the other. And most of them are random patterns that don't have any inherent interest or meaning. And within this stream, you have two critical targets, T1 so-called and T2. T1 is usually a simple detection task, it can be anything, but it's something that within the stream is very simple to detect. The participants are typically at 90% or higher in detecting T1. In this case, they were detecting that there were these faces. They were familiarized with 3 faces, Andy, Bill, and Chad, and they just had to say at the end of the 2 seconds, they had to say T1 was Andy. But they also had to report to us on this T2. The second target is the critical one, because if the second target follows the first one from around 200 to 400 milliseconds, there is a blink, an attentional blink. So it's almost as if the person is blinking their eyes and can't see it and misses it. But in this case, it's not a physical blink, it's actually an attentional blink. They're staring at the stimulus, not blinking, and just there's plenty of time here to detect it. But for some reason, perhaps still being worked out, given that the resources are consumed in processing this initial stimulus and putting it in a way that you can report it at the end of the trial, there is this attentional blink. So what happens is that in a large proportion of trials, this T2 is missed. The person is not aware, is not reporting that there was a scene, in this case, this kind of house. So what we did was, can we actually change this lack of detection, this difficulty in detection, by creating a T2 that is more potent? And by more potent, you could have it brighter, you could have it some other kind of moving thing. But what we did was actually to create an effective manipulation in which one of the kinds of T2 would become a conditioned stimulus, would be affectively significant, and the other one would not. So what we did was, before the task was this attentional blink. Before the task, we performed a conditioning phase in which participants saw, for instance, these houses, and there was houses were always paired with mild shock. This shock is ethically done. The participant sets the level of the shock so that it's actually not painful, but pretty unpleasant. And so every time that there's this house, they receive the shock. Every time that they see instead this outdoor scene, not with a house, but with a skyscraper, with a building, there was no shock. So there were these two kinds of T2, and they had to indicate whether the T2 was a house or building. So what we saw was that, in fact, behaviorally, this affective content on the T2 performance, on the performance of the attentional blink. Remember, the attentional blink is performed later. First, they do this conditioning phase. What we find is that if the stimulus is a CS minus, the blink is quite severe. They only detect around 63% of the time they're correct when they see outdoor buildings. But when they see houses, there is sort of, it counteracts. It's a stronger stimulus that you don't blink as much, attentionally blink so much. So you gain around 10% or so that your performance here is around 73, 74%. So behaviorally, we attained what we wanted to attain, which is having this emotional affective significance actually made your stimulus more potent, such that you would have less of an attentional blink. And I should say, which I should have said at the outset, during the experiment itself, there is no physical shock. These stimuli are only used during conditioning to create the affective significance. During the performance of the attentional blink, the shocks are not administered. So the stimuli are these houses and buildings, and they're counterbalanced across participants, some of whom receive shocks for the buildings and so on, but I'm giving the results here from the perspective of these houses. Okay, but what we were really interested in was really understanding the way in which something came up on my screen. The way in which the attentional blink, what are the circuits? How is this actually this competitive advantage of this emotional stimuli? How does it come about? So one of the things that we did was to perform this during scanning, and we knew that the amygdala is incredibly important for this conditioning manipulation that allows let's say the houses to be more effectively significant. So what we were interested in is looking at the relationship between responses in the amygdala on a trial-by-trial basis for each one of those trials of the attentional blink. The person, for instance, detected correctly T2, missed it, and so on. So we could take trial by trial and look at the activity level on a trial-by-trial basis. And what we saw, what we wanted to establish was its relationship to behavior. So we have the responses on the one hand, and we have the behavior. And what we found was that the higher the fMRI signal from the amygdala, the higher the likelihood that the person would detect the stimulus. So there is a suggestion then that for the affectively significant stimuli that evokes stronger responses in the amygdala, then the participant is actually detecting this more reliably. But what we wanted to do was to go one step further and understand, is the amygdala perhaps changing the way changing the way the visual system responds, so that responses are stronger in visual system for stronger stimuli, for affective stimuli, and that actually confers the attentional advantage that these stimuli have. So what we did was we looked at a part of visual cortex called the parahippocampal gyrus. And this parahippocampal gyrus has been characterized in the literature by many people. Many groups have shown this. I think the first results, I think, were from Nancy Canwisher and her group. I can't remember exactly now, but but it's well characterized that it responds very strongly to these stimuli. And what we saw is that when we looked again at trial by trial fluctuation, it was the same relationship. In trials in which the person was detecting the T2 reliably, the responses were stronger. And the idea then is that via anatomical connections that are known to exist between the amygdala and the visual, this part of visual cortex, there's an advantage to the responses of the effectively significant stimuli that allow the behavior to be improved for the CS plus category. And in fact, if we perform a mediation analysis, this relationship between amygdala and behavior is explained in this mediation sense, in this statistical sense, via this relationship via the visual cortex. Obviously, this is not something that we can make any causal statements. This was an fMRI. There was no perturbation or anything like that, but at least it's consistent with a notion that there's a circuit that actually, from the amygdala, stimulates enhances response in the visual cortex that favor the behavior of the CS plus stimulus. Okay, so at the same time, many investigators were proposing this type of effective processing that takes place and that was reliant on the amygdala. The amygdala is one of the key structures to allow this enhancement, as we illustrated in the previous study, is actually one that takes place in a largely automatic fashion. So Ledoux here was suggesting in his 96 book that, for instance, the person is trekking in the desert here and it's actually has the rattlesnake in the visual field, but is not paying attention to it, is not looking at it, but it's right on the line of sight. And in sort of an automatic fashion, then the responses activate the amygdala and allow this person to run away in a way that is completely automatic, suggesting to several people that this fear and fear learning centered on the amygdala really comprise an evolved module that is at the center of fears, phobias, and our preparedness for this kind of stimuli that are important in this argument for stimuli that have been important in an evolutionary sense. So what we did was, with Ralph Adolphs and myself, we analyzed the results of the literature, and we described the results in the following sense. There is a standard at the time, and to a large extent to this day, there's a standard hypothesis in which emotional stimuli are suggested to be processed by a sort of dedicated modular system that operates quite rapidly in an automatic fashion, no need for attention, and might be even functioning largely independently of conscious awareness. And important clinically, it's not just for basic research, that defects in this system underlie phobias, mood disorders, and PTSD, post-traumatic stress syndrome. And so it's not something that just is something that is a laboratory curiosity or a laboratory phenomenon, I should say, but it's something that actually has real-world consequences to conditions such as anxiety and mood disorders. So his group, separately from mine, and then we decided to write this paper, we started looking at sort of several cognitive emotion interactions. We had been looking, and we continued looking since the paper, at several types of interactions that we wanted to understand the extent to which these kinds of statements really held up. Is this really a modular system that operates in this manner here, this special manner? So what we did was that, is it operate independent of awareness? Does it operate independent of attention? Does it operate independent of the context in which the object is shown? So-called high-level factors that might affect certain kinds of processing, but that were suggested to not impact this kind of processing, this kind of special processing by emotional stimuli. So let me show you briefly one study that we did. This was a study that I did in Leslie Ungalaida's lab, and we did many variations that had better designs, but I'm just going to stick with this one because it was the first one, in which what we did was we had these faces shown to individuals during scanning, and there were two conditions. One in which the person was supposed to fixate the center of the face and indicate whether these faces were male or female. So it's a very simple male or female looking at, and so it's a very simple task. And in alternating blocks of trials, what we did was ask the participants to maintain fixation in the same place, in the center of the face, and just move their focus of attention to the periphery without moving their eyes. And we make the presentation time short so that it's not beneficial for them to move their eyes, because if they move their eyes by doing that, the stimulus is only shown for 200 milliseconds. And in later versions, you can use eye tracking, can do all sorts of kind of checks to make sure that the eyes are maintained in the exact position. But nonetheless, but the key part is that what we wanted to do was to have the person have the exact same visual stimulation, just move their attention to determine whether these peripheral bars were the same orientation or different orientations. If they're different orientation, or the same or different, is actually a task that is quite hard to do, because these are peripheral stimuli and they're calibrated so that people would have a really hard time doing this task. So what we found in this task was the following. We focused on the amygdala, and shown here as a region of interest, so that we extracted signals from the right amygdala in this case. And what we saw is that, as others had seen in the past, if there is a response to a face that is expressing fear, we find a strong response that is quite stronger than the neutral face, and we found some intermediate response to a happy face. So this is something was quite well known at the time. There was nothing. It was a replication. This part replicated prior findings. The question was that what happens when you are not attending to these faces, and you're actually doing the bar orientation task. During this unattended condition, what we found was in fact that there was no differential responses. They were kind of flat responses to these faces as a function of the type, fear versus neutral or happy in this case. So the bottom line from this study and many other studies that did this in several variants from my lab and from many other labs is that the effect of valence actually depends on attention. If the attentional manipulation is strong enough, then these valence effects, this advantage of the emotional content is not expressed, doesn't affect behavior in that way, and brain responses. We also did, just very quickly here, we also did experiments with in which we manipulated awareness. In this case, we did a masking task in which a mask is actually a face of another individual, and it's, in this case, for instance, it's a mask of this person having a neutral expression that masks either a fear response or a fear face or a neutral face. And so what we did was that we did this careful calibration with this. We can talk about it later, how we can do this. But essentially what we found was that when there was the conditions of the stimulation were such that the first stimulus is shown long enough that people are aware of them, we find these responses in the amygdala that are stronger for fear-containing versus neutral-containing stimuli. When people were unaware, which was if the first face was shown for only 33 milliseconds, then we did not see evidence for differential response. And this is not just a simple issue of thresholding. As you lower the threshold here, what we find is if you lower the threshold here, what we observe is that you see sort of like noise coming up in sort of all sorts of a salt and pepper unstructured way, and nothing that resembles sort of like a cluster of activation here in the dorsal parts of the amygdala. Okay, so what we conclude is that there is no question that affective processing is prioritized. We saw that in attentional blink. Many people have suggested that, but they also have suggested that it's prioritized to the extent they become this kind of almost all too powerful class of stimuli. They are processed without attention, without awareness. They're just this incredibly powerful kind of stimuli. And what we find is that given careful enough manipulations, we can actually find many settings in which these stimuli are not able to overcome the demands that are being presented, that are present. So essentially, their processing is also modulated by attention, in our hands by awareness, context. We also did contextual effects and other kinds of manipulations, suggesting that affective processing is indeed prioritized, but it's not strongly automatic in the typical traditional sense that many have proposed. Okay, so I don't know if there are any quick questions of clarification that you want to ask. I'm going to shift now to the motivational reward side of the equation to show you a study. I don't know if there's anything that was any pressing questions or concerns that I could address now. So otherwise, I will continue. So what we wanted to look at was to focus on a hypothesis that wanted to investigate, to address a hypothesis that wanted to investigate the extent to which motivation, as manipulated by cash rewards, because we're not doing primary rewards with humans, was a little tricky to maybe have some people developed ways of having juice and asking people to withhold from drinking for a few hours and have juice rewards, but we ended up using these secondary rewards that happen to motivate people quite a lot, which were the promise of cash reward. And the hypothesis was that motivation would enhance executive function in a way that was fairly selective, in a way that was not just some kind of generalized enhancement of arousal so that people would be really fast, or some response criterion change that people would be really careful. It was more that the effect would be selective, as I'm going to illustrate in the task in a couple of slides. So we did this in the context of cognitive control and asking if it would reduce response conflict. The kind of response conflict that we were studying was something that was very much like the Stroop task, the Stroop effect that is fairly well known to most of you, which in this case here is a task in which you might show a word, and this word it spells blue, but the color in which the word is written is red, so there's contrast, there's conflict between the two, and potentiates the response red and blue simultaneously. And the person is asked to report on the ink color, so in this case it's red, but the person is sort of also, with people who are schooled for decades, they read these, it's a very powerful type of manipulation, so the person is reading this in an almost obligatory fashion. You look at something, it's hard to not read the word. So what we did was we did a variation of this, again, by looking at response in visual cortex, because we wanted to kind of do some kind of analysis based on where these stimuli most strongly engage, so the perihippocampal gyrus, but I'm not going to discuss that here today. But that explains why we use a version of this Stroop task that it was determining whether these pictures were pictures of buildings or houses. And you could have these strings here, house or building, on top of them that were task irrelevant, so that they could create, like in the Stroop task, the congruent or incongruent case situations, where, for instance, here, obviously this is a building, but it's written house on top of it. So our trials were identical in structure, except that the person could receive quite nice reward at the end of the trial, at the end of the task, for performing these kinds of trials correctly, and in a way that was relatively fast, and in a control condition in which they did not obtain an extra reward except for the base pay that they're provided to participate in the study. And essentially just showing these kinds of stimuli as I outlined before. And yes, and so what we observed was in this kind of paradigm, a reward by cognition interaction, which was that the effect of interference of the incongruent condition was reduced with reward. And so the way we do that is actually by introducing a third condition that allows us to separate interference versus facilitation. So it's common in these kinds of tasks by having a third condition that doesn't interfere by slowing you down or doesn't speed you up, because the two things are not congruent or incongruent. It's just a separate category. So these X's here are so-called neutral condition. And what we find is that when we have the no reward condition, so in those trials, the incongruent trials are 50 or so milliseconds slower than the neutral ones. That's the usual slowing down from the interference that has been observed and is observed all the time with these kinds of tasks that is quite reliable. What we wanted to test, however, was what would happen with reward. With reward, two things happened. One was a generalized speeding up. People were on average around 50 or 60 milliseconds a little faster, so around 10% faster than they had been. So this was not, they were so fast, for instance, that there was a floor effect and all the conditions were equally fast. That was not the case, and we can do some kind of analysis to rule the kind of floor effect as well. But what we found was an interaction in which this interference effect here was reduced when in the face of the possibility of earning a reward. So we see a reward by cognition interaction in that this interference effect is reduced. So we did several analyses, and if you're interested, you can take a look at the paper to see how we looked at it in terms of a general the kinds of interactions that might be supporting this effect of minimizing the impact of the incongruent string or word in the reward condition. So how does that happen? But what I want to focus here today is a little bit more on the side of networks. And you will see how I'm going to develop this in a slide or two. So if we step back and we think, how does executive function might be interacting with reward? So the way we addressed this was to look at regions such as the frontal eye field here in frontal cortex, the intraparietal sulcus here in parietal cortex, that have been known by many studies, have been shown by many studies, showing that they're really important for attentional selection. And other studies, there's a large literature showing that regions, for instance, such as the ventral striatum here, are really important for reward-related processing. They have a lot of dopaminergic dopamine receptors. These regions receive signals from the midbrain, the dopaminergic midbrain. So what we were interested in looking at is, again, going back to the attentional blink, this fluctuation of these responses on a trial-by-trial basis. If we look at the response to the initial cue that indicates whether the person can receive reward or no reward, if we look at these responses on a trial-by-trial basis and look at their fluctuations, what we can now do is look at how this trial-by-trial fluctuation in these regions is related to the trial-by-trial fluctuation in these other regions. So these other regions are also fluctuating. We have signals for the entire brain that we're collecting with fMRI. And so we can look at the functional coordination or degree of coherence or synchrony or correlation between the signals in these regions that are known to be really important for attention and regions that are really important for reward. So what we found was that, I should say that more broadly, this idea of functional coordination is something that has been used in electrophysiology since the late 60s and in functional MRI since the 90s. And the idea of computing something called a correlation or functional connectivity by looking at the correlation between signals of two regions, irrespective of their anatomical connectivity, just to understand the functional relationship between these signals. And what we found was that the functional connectivity between these regions in parietal cortex that are important for attention and parts of the subcortex that are really important for reward actually substantially increased and significantly increased in reward trials compared to no reward trials. But this is something that had been in some fashion or another shown by many others, and what we wanted to do was to go beyond this possibility of linking just pairs of regions and looking if these pairs of regions had increased coupling or not. So what we turned to was to doing a simple network analysis, a graph theory kind of analysis, in which we had a series of brain regions that we outlined and created these regions of interest. These were regions that just expressed the simple effect that they responded to any cue at the beginning of the trial. The cues could be $20 reward or 0. They were responsive to these cues. So we took these as the regions of our network. So these are the brain regions. And we took the connections for this graph, for this network analysis. What we took, the strength of the connection between them was their functional connectivity, which in this case is the trial-by-trial correlation of the responses in these pairs of regions. So we can construct a network. And when we construct a network, we can look at the property of this network and how it sort of organizes itself. And because this is a high-dimensional kind of organization, you can visualize it by projecting in 2 dimensions by these kind of force layout algorithms that sort of spread out the nodes that are have higher links between themselves and separate them as much as possible in this when you project down into this 2-dimensional sheet. So you have, for instance, a cluster of units here that have more stronger connections between themselves and relatively weak connections with other parts of the network, and vice versa, correspondingly in other clusters here, this pink or this purple cluster. So what we found was that if we look at this set of regions that we outlined in the beginning, under the no reward condition, they form 2 clusters. They form these 2 clusters, and there's nothing inherently important or critical or essential, I should say, about these clusters. It's just one way in which the network kind of reorganizes itself, that there are a cluster of mostly cortical regions that happen to be important for cognitive control, and there's a set of regions here that are important for reward-related processing that have stronger functional connectivity within than between the clusters. But again, this was not really the aim of this study. The aim of the study was not to find out the exact way in which it was organized, because I don't really believe that. I think that there are many ways of looking at the organization of these complex systems. What we were really interested in was looking at instead how reward would change the pattern of functional connectivity in this network sense. And what we found was that when the reward cue appeared, what we saw was that there were these large-scale changes in connectivity, in functional connectivity, such that it's not appearing here the way it was appearing on my other computer. But in any case, what is showing here is that the nodes of this cluster here are much more strongly functionally connected with the nodes of this other cluster here under the reward condition compared to the no reward condition. So what is happening is that these clusters are much more functionally integrated during the reward, such that the system is then now less modular and more integrated during the reward versus the no reward condition. And you can quantify this and get p-values and things like that, computing specific indices of modularity or global efficiency. You can kind of quantify that in many different ways. But the point that I want to make here is that instead of focusing on pairwise connections, we can take a network perspective and look at the connections, for instance, from one region, the caudate on the right hemisphere, and see how it might be changing its connections to as many nodes as you have. You can investigate sort of like the changes that it actually, the way it reorganizes its functional connectivity as a function of the reward manipulation. Okay, so let me just provide you a quick example now in the domain. This was an example of these network interactions in the domain of, in the appetitive domain, in the reward domain. I want to go back now to the emotional affective domain, the threat or shock domain, the aversive domain, I should say. In the study that we performed, And we wanted to look at these kinds of interactions with a wider group of networks, I should say a wider group of regions that span more networks or a large set of networks in the brain. Remember that the one that we performed for the reward manipulation was a relatively limited number of regions that were responding to the cues. So we wanted to have a little bit more general analysis in the aversive domain in this new study. This new study was a very simple one in which people just stayed without moving their heads in the scanner and not thinking of anything in particular for a period of around 60 seconds. There was a variability between blocks. And one condition when they were in the safe block, it was basically essentially the resting state. The rest condition, they don't do anything, they just stay there, they're not shocked. During alternating or during other kinds of blocks, there were these threat blocks in which they could receive 0 to 4 shocks. So in these kinds of anxiety-producing blocks, sometimes there were as few as 0 shocks, and sometimes as many as 4 shocks per block. So we actually could focus on the blocks that had 0 shocks, but there was a potential shock. So in these blocks, what we're seeing is that what we have in these blocks are blocks that do not differ physically from each other, except that there's a difference in color of these signals, these symbols that indicate which block type you are in. But aside from that, they're just the same. And what we were interested in doing was doing an analysis of determining the overall modularity, or you can think of it in terms of network efficiency, which is how effectively can signals travel in a network, for instance, gossip in a social network, or signals in an anatomical network, in this case, in a functional network, because we're using the functional connectivity between the nodes as the strength of the connections between nodes. So what we did then was to consider these so-called large-scale networks that have been studied in the literature, the salience network, the default network, and the executive network. And we took a few representative nodes from these networks. And in the end, what we did was we took brain regions from these 3 major networks, the salience, the executive, and the default network. And we added 2 regions, the amygdala, which is important for affective processing, as well as this region called the bed nucleus of the stria terminalis, which is also it's less known, but it's very important for anxious apprehension and more sustained types of anxiety type of experiences. And so just in the interest of time here, just showing you very briefly, what we were really interested in was the kinds of changes that we would see in organization of these networks going from the safe state to the threat state. So again, there's nothing inherently fundamental about the brain that is like, we can recover its unique configuration during safe or threat. We view it as more, what are the differences that we can detect between these 2 states? And what we were doing is trying to look at these networks with this manipulation. So we see these networks, for instance, the salience network here shown in red, the nodes of the salience network shown in red, nodes of the executive network shown in green, and so on. So there is this change here. There's a substantial change from the safe to the threat, just visually here. But the goal is just to illustrate. Well, the important thing was that can we compute a series of metrics, network metrics, within network metrics, between network metrics, all sorts of interesting ways of looking at the organization of the system in terms of network science, and can we detect consistent changes in this organization? And just to highlight one finding that we found is that what we found was this decreased efficiency, the ability of information to presumably flow through the network, that was decreased in both the salience and in the default networks. So what is happening, one way to summarize is that what is happening during the sustained threat state that the person is undergoing in those blocks in which they can receive shock is that these signals in, for instance, the salience network and in the default network, they're becoming a little bit less coherent than in the safe condition. So again, as one way to understand how these affective or motivational manipulation might be having effects not only at the level of responses, localized, let's say in a task, in the Stroop-like task, or in the attentional blink task, but more in a system level, large scale level of looking at the system. So before I wrap up, I just want to remind you that many of us have been involved in thinking about naming these networks, and we have Nathan Sprang and Lucina Wooden have been spearheading a group to be thinking about this, and it's a hugely challenging endeavor to try to change things that are so ingrained already that people use salience, executive, default, and so on. But one of the things that I wanted to remind you is that we call these networks for convenience a salience network, an executive network, and so on. But these networks are involved in a lot more. Salience network has nodes, and as a network itself is involved in a lot more than just salience. And the default network is involved in a lot more than just autobiographical memory and self-related, self-relevant or related processing. I just want to convey the idea that we really should be looking at these networks as really multifunctional, not just giving it a label doesn't turn it into having a single function. It really is something that we have to study very carefully and look at how in different contexts these regions might be performing actually different functions. Okay, so let me wrap up here with some final thoughts. So what we see is that traditionally, both in anatomy, or I should say both in neuroscience as well as in psychology, there was, and to some extent there is, a trend of separating these domains, for instance, cognition and emotion, and even maybe to some extent having a separate motivation pathway that involves separable processes with separable circuits. My take is one in which we should approach these systems from a different perspective, one in which does not try to separate them, and in fact try to understand how they interact and their signals are integrated. But this actually poses a much more general question about how do we actually study the brain. If you take a popular textbook, let's say the Gazzaniga, Ivory, and Mengen Cognitive Neuroscience textbook, it's natural to organize its chapters in terms of perception, attention, memory, learning, language. Yes, there are some relationships between these things, but by and large, we study them as independent units. So the goal of neuroscience and study the brain seems to be learning about these mappings between these mental domains, perception, attention, cognition, and so on, and the neural underpinnings of them in a more or less systematic fashion. And what I would like to suggest, kind of also going back to that slide of the amygdala and anatomical connectivity, and taking inspiration now from the anatomy, not just of the amygdala or just in that case, but looking at the vertebrate neural anatomy and looking at it more generally and trying to understand how it might be organized in terms of large-scale systems, in which, for instance, the pallium here, which is the cortex in mammals, but with the corresponding area in birds and reptiles and amphibians and fishes. The pallium is interacting very heavily with the subpallium, subcortical areas. It has these massive connections with thalamus. The thalamus has massive connections with the midbrain and hypothalamus, essentially suggesting that we should look at this in terms of these large-scale connectivity systems and how this information flows in a more global sense, so that we can understand the kinds of behaviors that emerge from this in a way that doesn't isolate these mental domains in such a way as we have tended to do in the past. So what I would suggest is that we can think of the brain bases of cognition, emotion, motivation, if we think of them in those terms, at least as engaging these large-scale dynamical cortical-subcortical networks. And the brain architecture really provides this underpinning, the basis for the distribution and integration of signals that are related to perception, cognition, emotion, motivation, and action. So I'd like to suggest that even going forward, that one way to think about this is that we don't have to let go of everything that we used to do, but I think it's also beneficial to push ourselves a little forward and think that the vertebrate neuroarchitecture, especially in mammals and in birds, is such that it really doesn't respect these boundaries, or doesn't respect boundaries of these standard mental terms. So it's not as if you can go into the brain and point, emotion is here. It's in this part, plus this part, plus this part, plus whatever. You can add a few, subtract a few. If you have a different version in mind, that's fine. But it's not a really productive way to just point to and say that emotion is here and cognition is this other part, and so on and so forth. So what I would suggest is that we really should be focusing instead in kind of these models in which the integration between these faculties that we tend to separate is really at the core of what we want to understand. So instead of isolating cognition and understanding cognition faculties or emotional faculties or motivational faculties, we should really look at the integration and how they support complex behaviors. Because we are interested in behaviors, complex behaviors, animal behaviors, human behaviors. And so how are they actually supporting these behaviors? It circles back again to what kind of system is the brain? And the brain, we know, has evolved, or we suspect, the brain has evolved to provide adaptive responses so we can survive, right? To problems that living beings face in order to survive and reproduce. So with this in mind, what I would suggest is that we need accounts or ways of thinking or frameworks or even vocabulary that, contrary to the way we've been doing things in the past, which is using a standard account that tries to use language that tries to emphasize the independence and separation of things, that we should really adopt a language or create a language that is a language of interaction and integration at its core. And just to finalize here, just an example, if you're interested in going into this a little bit further in a more in-depth way in the domain of that is more closely related to emotion, I discussed this in a 2017 paper in Trends in Cognitive Sciences, and how we can think about this in terms of what I call functionally integrated systems that actually span the entire neural axis from cortex, subcortex, midbrain, brainstem, and here omitted here, but the cerebellum as well, in terms of a series of incredibly well organized and systematically organized. So it's not that everything connects to everything in an unsystematic fashion, quite the opposite, but there's this exuberant, massive amount of connectivity in a very organized fashion that we need to kind of, from there, understand how these functionally integrated systems arise to meet the behavioral demands of the tasks or the problems that animals face for survival. So in a more user-friendly or more general audience kind of view is something that I also outlined, as Klaus was mentioning, in this recent book that I published, that is very much inspired by this way of thinking that tries to look at how all of these mental domains are really interrelated to the extent that it makes it for something that is entangled, not in the sense that you can disentangle them, but in a way that we should be trying to understand what this integration means in terms of our capabilities and our deficiencies in mental health and mental disease and so on. So just to conclude, I would like to, this is research that I did over many decades with many graduate students and postdocs and many colleagues, a lot of engineers and research assistants that I'm very grateful for the many years of research any one of these studies took, and incredibly fortunate to be funded by the National Institute of Mental Health that funds this research. And as Klaus mentioned, if you're interested, I'm somewhat active on Twitter, and I try to keep that activity to scientific questions and things about the brain. Thank you very much.
Thank you very much, Luiz. I'm doing some clapping here on behalf of the audience, but you see more of the clapping hands also there from the other members.
All right, thank you.
Thank you. That was beautiful, and that was very clearly structured, and it was a great lecture telling us how emotion and motivation, which are subjects that not so many of us are working on, can be straightforwardly integrated in a network perspective. And so I think that's really beautifully instructive.